So good morning also from my side. It's my pleasure to chair the next session of short talks. We will have only three short talks now and then have a break. And the first speaker is Charlotte Soneson from Basel, from Switzerland. And she will talk about single molecular footprinting.
Thanks, Robert. So today I'd like to introduce you to footprintR, which is our package for representation and analysis of single molecule footprinting data. If I can move this forward. Yes. So this is really a team effort, right? So this is the rest of the team. Michael is also here. We have Panayotis, Sebastian, Dirk, Elisa, and Hervé also contributing very much to this project. So I just want to make that clear from the start. Single molecule footprinting allows us to get a very high resolution readout of chromatin accessibility. And the way this works is that you're basically taking your nuclei, you're incubating them with an exogenous methyltransferase. And there are many of these methyltransferases. The data you will see today was generated using EcoG2, which is a sequence-agnostic adenine methyltransferase, which basically means that it will go into the cells, into the nuclei, and methylate every adenine that it can access, regardless of the sequence context. So that means that after this step, all the accessible adenines will be methylated, and all the inaccessible adenines, which could be adenines that are bound by a transcription factor or that are inside the nucleosome, they will be unmethylated. So somehow we need to read out this information to make inference about the accessibility, and we do that using nanopore sequencing, which gives us both the nucleotide sequence as well as information about the modification of each nucleotide. And of course, if we look at the modifications of the adenines, that will tell us something about the accessibility through this working of the EcoG2. But so we work on mouse embryonic stem cells, and they also have endogenous DNA methylation, right? And that will happen in CpG contexts. So if we take our nanopore sequencing data and we read out the modification of the CpGs, we also have a readout of the endogenous DNA methylation of the same reads in the same molecules. So in a way, we kind of get two assays at the price of one. And if you look at the data, whoops, that was not good. Can I go back? No. We cannot look at the data. So, in any way, what you would see here is that you would have basically two readouts, right? The accessibility and the DNA methylation. Yeah. Anyway. Sorry? We have to draw it, but there's no screen. There's no whiteboard. Okay, let's try again. No. I wonder if there are any other images in this. Huh. Interesting. Okay. Let's just talk about it. Okay. I had the slides. I had the figures in my PowerPoint. I have it on my laptop. Yes. So
shall we maybe switch to another talk first? So in order to make it smooth, maybe we can switch to the next talk. So this would be given by Ning Shen, who is coming from overseas, and would be talking also about DNA methylation, about deconvolution of the signal from the immunoprecipitated assay from the MEDIP-seq. And please come to stage.
Okay. Hi, everyone. I'm here to introduce one of my recent work, which is developing a cell type deconvolution method for methylomes, which is DNA methylation immunoprecipitation-based sequencing method. So just a little bit of a recap. DNA methylation usually happens on the cytosines, and most likely cytosines in a CpG site. It is one of the most important epigenetic markers. It's important because it governs gene regulation and influence cell identities or cell functions and genomic stability. So aberrant DNA methylation patterns have been associated with a lot of diseases, including cancer and neurodevelopmental disorders, etc. So one of the motivating examples of our method is to understand the components of cell-free DNA in the context of a liquid biopsy experiment. methylomes is one of the protocols that's suitable for this kind of application because it's effective. It requires low input DNA to be effective, and it's also very cheap, and it also provides genome-wide coverage. So cell type decomposition is very essential for this kind of data because in the liquid biopsy example, these cfDNA fragments, they might come from a lot of sources, such as tumor cells, the immune cells, or blood cells. So our solution to understand this data is cell type decomposition, which tries to estimate the cell type proportion from the bulk data, and these cell type proportions can be used for adjust for the cell type specific signals or enable accurate disease detection even. So a typical reference-based cell type decomposition algorithm look like this. You have the genomic data, you have you have the reference matrix that's independent from the data, and then you use this reference matrix to infer the cell type decomposition in the specific samples you're interested in. And then with the estimated proportions, you may compare, for example, do group comparison in the downstream analysis. However, this methylomes technology has been around for quite a while, and a lot of people are using it for liquid biopsy, but there has been no cell type decomposition algorithm that's developed for it because it's very challenging. Well, first of all, methylomes measures the relative enrichment instead of the percentage methylation, such as microarray or whole genome bisulfite sequencing. This is just an illustration of how the methylomes signals look like. And also it's sort of biased to the CpG density. And also it only measures the methylated regions. It doesn't know where the unmethylated region, because the antibody just binds to the regions where it's methylated. And it's very noisy and sparse because most of the times we're dealing with cell-free data. And last but not least, we don't have any methylomes specific reference atlases. For most of the previous algorithm that's developed for cell type deconvolution, for array, they have array atlas. From WGBS, they have WGBS reference panels. But we don't have that, so we're going to just use the panels from the WGBS or arrays, which makes this development more challenging. So we wrote up this paper. We called this method decemedip. And it uses a Bayesian hierarchical model to do this estimation of the cell type proportions. And this model composes two main parts. Well, the first part is the logic normal component that models the cell type proportion. And the second part is the GAM component that models the relationship between the methylomes read coverage and the percentage DNA methylation level from the reference panel. And each of these components doesn't work separately because we don't have that information, but we combine them into a Bayesian hierarchical model, and we can estimate these parameters simultaneously. So the input to the model is CpG density, of course, and then methylation levels from the reference panel, and also the methyl counts from the sample of interest. So this algorithm does estimation for each of the samples independently. We also included, we also included these anchor CpGs, which are CpGs that shows extreme methylation values across all the tissues to stabilize the MC-MC draws of the Bayesian model. So the output would be, of course, deconvoluted cell type proportions, and also it will spit out the translation pattern between the metapseed counts and fractional methylation from whichever reference panel you're using. So in the results, we sort of validated this model selection of the GAM component because you have a lot of choices to make in this GAM model. So GAM is, generalized additive modeling. And we see that, so we're using paired whole genome bisulfite sequencing and metapseq data from cell lines, which are considered to be stable across experimental conditions. And we're sort of using them as X and Ys, and then model this, fit this GAM model on the X and Ys. And here we're seeing that the orange dots are the original counts from the CpGs in the reference panel, and then the black dots are the predictions. So most of the times the posterior credible interval is covering the original counts, and it seems like the prediction is doing pretty well. So this validates the modeling choice. And then we ran a bunch of benchmarking experiments with simulated data. So here we were using a synthetic mixture data from fully generated by the model itself, because Bayesian models are generative models. And we were interested in coverage, cell type, and target proportion, etc. But what we see is a lot of times, these predictions of the proportion estimates are like doing a little bit of underestimating, which might be due to the correlation between the cell type, between the methylation markers of the cell types that causes a little bit of multicollinearity in the model, and we're already sort of adjusting for that in terms of introducing a correlation matrix prior, but due to the existence of the anchor CpGs, the correlation is a little bit higher than what we expected. So we also applied it to a large cohort of multi-cancer patients with plasma cell-free DNA measured. In this figure, we're showing the estimated non-blood proportion. So the healthy patients here, they usually don't have a lot of non-blood cell-free DNA in the bloodstream. But all the other cancer patients, except hepatocellular cancer patients, the other cancers are showing significantly higher proportion of the non-blood cells in the cell-free DNA methylome. And to zoom in to like specific cell types to zoom in to like proportion of specific cell types, we see that the cell types that's corresponded to the cancer, for example, the column cells corresponding to a colorectal cancer, they have a very high proportion, a very high deconvoluted proportion compared to the other samples. Okay, so in the same data set, we also have this valuable information of circulating tumor DNA content, which was measured by low-pass whole genome sequencing, which is independent from the metapseq measurements. And from here, we can see that the content of ctDNA is very much positively correlated with the estimated proportions of either the non-blood cells or the specific cancer tissue types of interest. So these are the results that I was showing you was partial. More are in the paper, but the key contribution of this framework is that it's the first method to deconvolute the enrichment-based data, not only methylomes but also others. And also it's first method to integrate the reference methylation data from alternative platforms such as WGBS or microarrays. And also we use a Bayesian modeling, so it provides posterior credible intervals we can use as uncertainty measurements. And also it counts for CpG density, which was a key cofactor, and it can be used to address the density-related issues. And the paper is in the archive, whereas it's currently under review, and we already submitted a package to Bioconductor. That's it. I wanted to thank all my colleagues in the Cord Hauer lab and my collaborators at Dana-Farber Cancer Institute. Thank you.
So we would have some time for a question. Is there any question from the audience? Then maybe I will start. Can you use it also for some other type of methylation assays, which are used for which are essentially pulled down by the enrichment? I don't know, it's like MBD.
I think it's quite straightforward to adapt this to MBD-seq. Yeah, but because they're a little bit different, so the model you might need to adjust the model components a little bit.
You need to tweak it a bit.
Yeah.
Hi, thank you for your talk. Maybe a detailed question, but very interesting is that you take into account the CpG density to remove the bias. So could you maybe give a hint on where in the model this is taken into account?
Yes. So this is like a generative process of the model. We specifically included in the part where the GAM model was fitting. So the D is a measurement of the CpG density, and we're using. Well, first of all, we're putting splines on it because the pattern is sort of flexible. And also, we're taking account into interaction between the CpG density and the methylation level in the reference panel. So it's a bit complicated. Yeah, I think one of the, sorry. Okay, too far. Like in here, you can basically see that each panel is CpG density, and then you see a little bit of differences of the relationship between the methylomes and WGBS for different CpG density values. I hope that makes sense.
Are there any other questions? If not, then let's thank Ning Shen again. And I will ask Charlotte to try the presentation.
Yes. Okay, we were here, right? So we have our single molecule footprinting data, and we concluded that we have both accessibility information and DNA methylation information, and this is what the data looks like. So here we have a small region of the genome. On the x-axis here is the genomic position. On the left you have the accessibility data. On the right you have the DNA methylation data. Each row in the top panel is a read or a molecule, and each circle is an observation. So first of all, we can see that the accessibility data is much more highly resolved than the DNA methylation data, right? Because we have many more A's than CpGs in the genome. We can also see that we actually have data from both strands. So can I point to this one? So here, these reads here are from one strand, and these reads here are from the other strand, because if we have an A in a certain position on one strand, we will not have an A in the same position on the other strand. And finally, we can notice that there are some regions here where most of the molecules are kind of inaccessible or lowly methylated, and then there are some regions here where instead most of the reads here or most of the molecules are accessible. And we can see that also here in the summary track, which is just aggregating over all the reads. So in footprintR, we provide a way to read this data straight from the standard output of the Oxford Nanopore base caller. And the way we store it internally is using a summarized experiment object. So this is what you see here. We have multiple assays. So we have summary level assays. So they have positions in the rows and samples in the columns. And these can store things like the coverage or the average fraction of methylation across these reads. But we also have read level assay, and here we have to be a little bit more creative because we have our samples and we have multiple reads per sample, right? So what we did was that we decided to store this in a nested data frame. So in each column in the data frame corresponds to one sample, and each of these columns is like a sparse matrix, or a special type of sparse matrix, namely the NA matrix, which is a nice kind of new type of sparse matrix that Hervé kindly implemented with this kind of use case in mind. And if anyone is interested in discussing more in detail, just get in touch afterwards. So we can read the data, we can store it. So what can we do with it afterwards, right? And at the moment, the methods that are implemented in footprintR can kind of be divided into two categories. So if you already know which loci you're interested in, so that may be CpG islands or promoters or transcription factor binding sites, you can kind of take a locus-specific approach. But we also implemented a genome-wide scanning approach where you can kind of look for regions that show a certain pattern of interest that you can define yourself. And I will just show one example of each of them just to illustrate what can be done. So here is an illustration of the locus-specific analysis. So here we have a 1.3 kb region around a CTCF site, so that's this gray thing here. And what we did was that we took all the reads that overlap this region and we split it by allele. So we have, I think, 13 heterozygous SNPs in this region. 2 of them are inside the binding sites. And for each read now, based on the sequence of these heterozygous SNPs, we split them into 2 groups. And the question then is there evidence for allele-specific binding of this transcription factor? So does the transcription factor bind to one allele and not to the other? And if we look, so these are the reads from the reference allele, and these are the ones from the alternative allele. And if we look here, we can kind of see evidence of these characteristic patterns of increased accessibility just flanking the binding sites. And then a decrease in accessibility exactly on the motifs, right, which is where the transcription factor is binding. So this would suggest that we have, actually the transcription factor is bound here, but here we don't see any of these patterns. So here it does seem that there is some suggestion that this transcription factor binds in an allele-specific way in this locus. For the kind of genome-wide scanning framework, the way we decided to implement this, I will not go into detail exactly how this is implemented, but we decided to make it in a modular fashion so that you can basically scan for any pattern that you're interested in, as long as you can provide a scoring function that kind of takes a genomic window and calculates some kind of score that says how much of this pattern that you're interested in that you can find in this window. So we implemented several of these scoring functions, but you can also provide your own. And the example I wanted to give was that we looked for regions that are differentially accessible, so that showed a different methylation rate in 2 different conditions that we were looking at. And what we're doing here is basically to bin the genome into small windows. For each of these windows, we're doing a fairly standard differential methylation analysis. We're getting a score, which in our case is proportional to the log of the p-value in a signed fashion so that you know if it goes up or down. And then once you have calculated those, you can do post-processing, like smoothing these scores a bit along the genome, thresholding them to only keep windows that that actually show some evidence of this particular signal. And then you can also optionally fuse neighboring windows that have a high score to get the actual regions that seems to change. And if you look at what can come out of this, so these are two of the regions that we found in the scanning. So this is one and this is another one. Here you have the reads from one condition, here is the reads from another condition. These gray boxes here are the differentially accessible regions that we found in this locus. And you can kind of see that both if you look on the read level data and if you look on the summary level data, in these gray boxes there is a lot more methylation in the second condition than in the first. So this is just one example. We can scan for many other things as well, depending on what we're kind of interested in. So with this, I'd like to round off. So I've shown you footprintR, which kind of provides this framework for working with single molecule footprinting data. We can import the data from the nanopore output files. We have this internal representation based on the summarized experiment and the NA matrix for the read level data. We can do visualization, we can do analysis. The development is still ongoing, so if anyone has feedback, it's very welcome. You can reach the documentation here. And we also started putting together an online book that kind of shows how to use this for real data analysis on actual data. So with that, I'd again like to thank the people involved, and I'm happy to take questions.
So are there any questions for Charlotte?
So do you think that the example that you saw for the CTCF, that this is a reorganization of topological domains around this site? Because the changes are not just on that position, but basically in the whole region.
Actually, so there are two mutations inside the binding sites. So that could be the reason that it actually, it's just the motif is not, the matching to the motif is not good enough for the reference allele, even inside these two SNPs here. But we haven't looked into more detail.
Is there any other question? I would have maybe one. So how easy is to work with because I can imagine you can have it like almost whole genome, like the data. So how easy is to work with that?
So we don't, in this analysis, we don't read the whole genome at the same time. So if you know which region, which locus you want to look at, you would read the data from that region, and that is pretty fast, and then you can just visualize that. For the scanning, we also read the data from blocks at a time, so we don't actually read all the data at the same time. I think you probably could, but the question is if that would really help.
So if there are no more questions, so let's thank Charlotte again. And the last speaker of the session is Jiayi Wang, I think from University of Zurich, and she will talk about the ATAC-seq data analysis.
So, hello everyone, my name is Jiayi, and again, I'm a PhD student from University of Zurich. However, actually today I'm presenting my master thesis project about bias correction and differential motif activity in ATAC-seq data. So I will start by dissecting my title. So first, what is ATAC-seq? The rationale of ATAC-seq is to sequence the open chromatin regions. In ATAC-seq, the genomic DNA is exposed to TM5, which is a highly active transposis, and TM5 will fragment DNA, and it inserts preferentially to the open chromatin regions, and then add the sequencing primers to the fragments. Therefore, the chromatin accessibility can be measured by simply counting the fragments overlapping with the region. And the region we are interested in is called peaks. which is the identified highly accessible regions. And therefore, more fragments overlapping with the peak, which means the peak is more accessible, and it's therefore more likely to be bound by the transcription factors. And the second thing we need to know from the title is about the bias correction. What are the possible biases in ATAC-seq experimental procedure, and why do we need bias correction? So here we're looking at 3 possible technical biases: GC content bias, fragment length bias, and the enrichment bias. So GC content is the proportion of G and C nucleotide in a genomic region, and it is a known bias also in chip-seq and RA-seq. So RA tend to amplify the GC-reached region less efficiently compared to the AT-reached regions. And at the same time, the most accessible regions themselves are GC content reached. So we expect a correlation between the GC content and the accessibility. More importantly, there are already studies, they reported that the GC content bias will impact the downstream analysis, such as the differential accessibility analysis. And then we have the fragment length bias. So the fragment length, they also decrease the amplification efficiency. So shorter fragments will have higher amplification efficiency. And also the longer fragments, they produce lower base quality and higher error rate in the second read in pair-end sequencing. And more specifically, in ATAC-seq, we expect a specific pattern of the fragment length distribution. So we will expect peaks at 100, 200, and 400 BP, which represents the nucleosome-free, mononucleosome, and the dinucleosome regions, respectively. However, the exact size will vary across the samples. And lastly, we have the enrichment bias, which refers to the variations in the degree to which the reads are concentrated in the high accessibility regions. So in chip-seq, this could be caused by the differences in the immunoprecipitation efficiencies. And in ATAC-seq, this could be caused by several reasons. For example, the ratio of TN5 transposes to nuclear ratio, and also the sequencing flow cell density. And altogether, these technical biases will complicate the downstream analysis. That's why we need the bias correction. And here we are focusing on the differential motif activity analysis. So here comes the question, what exactly is the differential motif activity analysis? So profiling transcription factor activity is important for understanding the gene expression and also the transcriptional regulation mechanism. And as I already introduced, with ATAC-seq, we can measure the peak accessibility. And then with motif matching, we can know that which transcription factor motifs are in which peaks. And therefore, we can construct an activity matrix for the transcription factors or their motifs by simply, for example, counting the fragments of peaks containing a certain motif. And then we can compare the activity across different conditions to infer the activity changes. So this can be simply done by adapting RA-seq tools like Lima or HR, and there are also specific tools designed for this goal. However, we realized that in those tools, the technical bias correction is suboptimal there. So that's why the goal of our project is to have a better bias correction method. Thus, we can improve the performance in the differential motif activity analysis. So the method we developed, we call it the weight model, because the main idea is to assign weights to fragments and peaks to remove the technical bias. So first, we have the fragment weights. This is based on fragments GC content and fragment length. So we split the fragment GC content and the fragment length distribution into bins by quantile binning, and therefore each fragment can be assigned to a GC FL bin, which is a two-dimensional bin. And then for each sample and each bin, we assign the weights to those fragments so that we can bring the distribution of each sample to their mean distribution. And then we got these weighted fragments so that we use those weighted fragments to construct a count matrix for the peak accessibility. And then we calculate we calculate a weight also for each peak, as that is to remove the enrichment bias I have mentioned before. So this is done by a nonlinear Loess-based normalization method called cyclic Loess. So for each pair of samples, we created an MA plot. So each dot is a tested peak, and on the M value on the y-axis is the log-fold change in accessibility between those two samples. And the A value is average accessibility in accessibility of those two samples. Because usually we only assume a small proportion of peaks will have the differential activity, so that the log-fold change should be centered around 0. So any upwards or downwards shift should be seen as technical bias. And with the MA plot, we fit a Loess curve on this plot, and so that we can assign a normalization factor on each peak. And this process is repeated on each pairwise combination of the samples. And finally, we evaluated our method. So we evaluated this on 9 data sets. And in each data set, we have one transcription factor we perturbed either by agonist or by knockdown or knockout. And then we expect a good method should rank this perturbed transcription factor as top as possible, and it should also give large absolute t values. And on the y-axis, we have all the methods we tested. So the baseline is just directly adapting the Limavo method. And in the red box, we have the best method in the recent benchmark paper. And lastly, in the blue box, we have our weight model, which performed the best since it assigns the best rank across all the data sets. However, we have 2 data sets. I think the perturbation does not really work well, so there is no signal to start with. And we can also improve the t-value in some data sets. So lastly, this is still a project in progress, and we actually have another standalone model called the insertion model, which we incorporate the transcription factor footprint information. However, because of the time reason, I couldn't talk about it today. And we are also testing on more data sets, so we find another 60 data sets which also have one perturbed transcription factor in each data set. And also, we are trying to wrap this as an R package so that more users can benefit from this. And at the end of my presentation, I would like to thank my advisors, Pierre-Luc, Emmanuel, and Mark. And also, I would like to thank all the members from the Robinson Group. Thanks for their feedback and their help. Thanks.
Thank you. So we'll have some time for a question.
Thank you. Very, very nice work. I'm wondering about these technical biases. You already mentioned that for GC bias, they can often be confounded with biology. Also the fragment length, for example. So what is your intuition? How does the method deal with not removing too much of these differences between samples that could reduce the signal?
I think because we're just trying to we bring the distribution between different samples to the mean distribution, so I think we're just removing the biases between the samples. And we also actually tested in a simulation data set where we insert the only the GC content bias and no technical bias. And we also tried to do it in an MA plot, but the MA plot is binning by the GC content of the peaks. And so I think that's how we tested. We only removed the GC content bias, but not the real biological biases.
Any other burning question? So if not, I would suggest to thank the speaker again, and actually also to thank to the other two speakers. And now is the time for a coffee break.